Hello, everyone. How are you? Thank you for joining again. This is lecture number 6 of the BICD140 Immunology course. So I would like to start today with a little a poll question to see how much you have been studying and fixing the concepts that we covered in the previous lectures. So I will start with this polling and now launch the polling, and so you should be able to see it. And so as always, I'll give you a minute or two to answer yourself, and then we'll go through the poll question. And of course, feel free to unmute yourself and ask me if it is not clear any of the answers at the end. Okay, so we end the poll here and we'll go through the answers. I'll end it. And so most of you answer E, as you can see here in the red. And so basically, so the statements ask for the true answers. So immunoglobulin M is normally pentameric and therefore not abundant in the extracellular spaces, because that makes the pentameric IgM bulky, and so is abundant in the serum and plasma, but not in the extracellular spaces because it's bulky to extravasate. At a single time, so this is true, B says at a single time, each B cell produces antibodies with the same antigen specificity. And so that's true because this is the antigens, the antibodies are produced from the BCR that is released in a soluble form. And they are always having the same antigen binding site than the BCR, and will always produce at a given time antibodies with the same antigen specificity. So that's true as well. C says antibodies maintain their specificity but may change their biological activity, class switching or isotype switching. And so we mentioned in the previous lecture that the biological activity of the antibody is determined by the constant region of the heavy change. And that constant region of the heavy change can be switched by a process that is class switching or isotype switching that we will learn more at the molecular level today. And so this is also true.
What would be highest spot for our meeting tonight?
Say again?
Yeah.
Can you repeat that? Sorry. Hello, Tom?
Sorry, I was unmuted.
Okay, yes. Please make sure you are muted unless you have a question for the course. So this says infection with extracellular parasites may be a reason for elevated IgE. And so we learn how IgE is important to fight these big helminth extracellular parasites because it binds to the Fc epsilon receptors in eosinophils, mast cells, basophils, that upon cross-linking of the IgE and Fc epsilon receptor will induce a signaling that promotes the release of granules that will produce histamine and lipid mediators that promote muscle construction to eliminate these parasites, and also some basic proteins that will destruct the parasite membrane. So all of them are true. So I'm glad that you guys have been studying this. So, okay, let's do one more and then we'll start with the content for today. So I will relaunch the poll like this, and then you guys can start. Okay, so I will end the poll here and share the results, and you can see most of you answer C, which is the correct answer. And so it says, which of the following statements about the antibody is false? Each antibody has 2 identical heavy chains and 2 identical light chains. So that's correct. So we learn about the structures of the antibodies, and they have 2 identical heavy chains and 2 identical light chains. In contrast to the BCR, an antibody does not have a transmembrane region, and that's correct. That's the only difference between the BCR and the antibody. The antibody, because it doesn't have the transmembrane region, now it can be released and secreted. Each light chain is composed of 2 variable regions. So that's incorrect. Each light chain, and again, there are 2 identical light chains, and each light chain has a variable region and a constant region. And the variable region of the light chain, together with the variable region of the constant chain, will form one of the antigen binding sites of the antibody. And the other one will be an identical antigen binding site that will be constituted by the identical heavy chains and identical light chains on the other side. So, so this is false then. So D says each heavy chain is composed of a variable region and a constant region, and this is correct. Okay, so let's go through the topics we will cover today. We will learn today that there are 4 mechanisms to generate antibody diversity, and we will learn how this works. We will learn on how isotype switching occurs and work at the molecular level. And finally, how do B cells secrete immunoglobulin or secrete their BCR, right? Without the transmembrane region, which is an immunoglobulin. Okay, so first, let's look at these 4 mechanisms that determine the diversity of the immunoglobulins. But before going into that, let's review a little bit the composition of the antibodies that we learn, and these poll questions were for that purpose as well. But just a refresher here, we learned that there are many constant regions in the heavy chains, that there are actually 9 different constant regions of the heavy chains in humans that form 5 different isotypes: IgM, IgG, IgA, IgD, and IgE, and some of them have some subclasses, so that's why it adds to 9 different constant regions of the heavy chains. We learn that this constant region of the heavy chain is what gives the antibody the biological activity, and so it's very relevant, the constant region of the heavy chain. We also learn, and this will be more relevant for the next lecture, but there are also several constant regions of the light chain, which in total are 5 in humans, and they have nothing to do with the biological activity of the antibody, okay? So the biological activity of the antibody is the constant region of the heavy chain, what determines that. And then we also learn that there are the variable regions of the heavy chain and the variable region of the light chain, which are forming the antigen binding site, will give the specificity to this particular antibody. And we learned that they are millions and billions of specificity because they are formed by these variable regions. But we'll go into more details on how these diversity in the variable region, in these antigen binding sites, are generated. How do we generate these millions of specificities? And so that's part of what we'll learn today. We will also learn on how we switch among constant regions of the heavy chain. Okay, so how is it possible to generate so many different BCRs or antibody with this diversity of the antibody, particularly in the variable region we will focus first. And there are 4 mechanisms for generating this antibody diversity. So 3 of them occurs before the antigen stimulation. And again, we are talking about here diversity in the variable region, right? So 3 of them occur before the antigen stimulation. And we will learn these 3. And then the 4th one occurs after antigen stimulation. And we will learn some facts about this one, but this one will be mostly fixed in a later lecture after the first exam, okay? The first 3 that occurs before antigen stimulation are the somatic recombination of different segments that encodes the immunoglobulin variable region, the precise recombination junctions. So this is variability in the joints of the recombined gene segments. And the 3rd one is the combinatorial pairing, that is the pairing of different combinations of immunoglobulin heavy and light chain. And the 4th one that occurs after antigen stimulation is called somatic hypermutation, and is a mechanism for introducing mutations, point mutations in the variable region of activated B cells, okay? So 1, 2, 3 is before antigen stimulation. Okay, so let's focus on number 1 first, that we said is somatic recombination of different gene segments. But first, to understand that, we need to learn a little bit of how is the heavy and light chain loci. And so here is the loci for the lambda light chain locus. Here is the locus for the kappa light chain. And here is the locus for the heavy chain, okay? And so the immunoglobulin heavy chain and light chain loci consist of family of gene segments that are encoding alternative versions of the immunoglobulin variable regions. And also they encode different subclasses of the constant regions of the immunoglobulin. So let's see, for example, for these are these alternative versions of that encodes for the variable regions are these ones here listed, B, J, and D. And so we have here, for example, in the heavy chain locus, these alternative versions that encodes for the variable regions. We have different alternative versions for the V region. Again, these are all segments of pieces of the variable regions. And so we have different versions for the V regions. We have different versions for the D regions, particularly 27, for example, here. And we have different versions of the J region, okay? So all of these encode pieces of that when combined, 1B, 1D, and 1J together will form the variable region, in this case, of the heavy chain locus, okay? This, in the case of the light chain locus, the alternative versions that encode the variable regions are B and J. There's no D there. And you can see here there are 40 in the kappa locus. There are 40 alternative versions for the B region, and there are 5 alternative versions for the J region, okay? And so when one of the V is combined with one of the J, will form the VJ, that together, these two now, that was one of these V and one of these J's, will form a VJ that now will encode for the variable region of the kappa change, of this immunoglobulin light change. And the same is for the lambda light change. The light change locus only have V and J alternative versions that encode for their variable region, okay? And then they also have, in the same loci, you have encoded subclasses of the immunoglobulin C region. So these are the different constant regions of classes of constant regions for the lambda change. This is the kappa has only one constant region here, and it's not all the loci here, but here you have all the constant regions for the heavy change. So C mu is the constant region for the immunoglobulin M. We have C delta is the constant region for the immunoglobulin D. And then you will see after that you have also constant regions for immunoglobulin A, and downstream you have constant regions for immunoglobulin G as well, and we will learn more about that, but know that this downstream, the C, the constant regions for M are the other constant regions are encoded as well here in the immunoglobulin heavy change locus, okay? So we'll read again these statements. So the immunoglobulin heavy and light change loci consists of family of gene segments. Each of these is a gene segment, right? Encoding alternative versions of parts of the immunoglobulin variable region. So this would be one segment that encodes an alternative version of a little part of the variable region, and different subclasses of the constant regions. Okay, so the variable regions are encoding by 2 or 3 segments that must rearrange before they can be transcribed. And so these variable regions for the immunoglobulin light change is encoded by 2 segments, the V and the J segment. And this needs to be rearranged, so brought together, one V and one J, for the variable region of the light change to be transcribed. And in the case of the heavy change, the variable region of the heavy change is encoded in this case by 3 segments, V, D, and J, and one V, one D, and one J needs to be rearranged and brought together to then be transcribed into what would be the messenger that encodes for the variable region of the heavy change. It's complicated, so let's see if it becomes a little bit more clear in the next few slides. So all these rearrangements of the different segments that encodes for the variable regions of the light change, B and J, remember now it's simplified here, but we have multiple V versions and multiple version of the J segment, okay? As we saw here, multiple, here particularly 30 different versions of the V segment, and here we have 1, 2, 4, and it could be more of the J segment. And so here is simplified, but basically that is to show that now one, for the light change to be transcribed, one V needs to be selected, one version of the V needs to be selected and rearranged, brought together with one version of the J segments. And this will lead to VJ recombination that occurs at the DNA level, okay? And that then only when these two are together can now be transcribed into RNA that will encode for the polypeptide that form the variable region of the light change. And this will be also here via splicing will be brought together with the constant region part of this polypeptide. Okay, in the case of the heavy change, this is again simplified. So first, there are multiple versions of the V segments, multiple versions of the D segment, and multiple versions of the J segment in the loci, in the germline DNA loci that for the heavy change. And we saw here this. There are 65 V versions, there are 27 D versions, and there are 6 J versions. Of course, I don't need you to remember the number of segments for each loci. Just know that there are multiple ones, right? And so this is simplified here, but again, this is just to show that this is the germline. But now in a developing B cells, for the variable region of the heavy change to be transcribed, one V needs to be selected and brought together by gene rearrangement first it will be a rearrangement of one D and one J that are brought together. And after this happens, then afterwards, this DJ is brought together with a V. And this is, again, by gene rearrangement. And this is all at the DNA level, okay? I highlighted this in red so you realize that this is all occurring at the DNA level, okay, in the developing B cells. So all cells have the germline, but only the developing B cells that are trying to form their VCR with the variable regions will have these, again, first the DJ rearrangements and then the BDJ rearrangement in the heavy change. And this, now after this occurs, now the a variable region of the heavy change can be transcribed and by splicing is brought together close to the constant region of this polypeptide. And we will see how the first one that is always produced are C mu and C delta. And we will learn that in the next few slides. But you can see how now these segments that have been selected will determine the amino acids that are now here in the variable region of the heavy change and the variable region of the light change. Is that clear? Let me go through one more slide and then I pause and make sure you ask all your questions, okay? So the V region encoded in the case of the light change, again, is encoded by V and J segments. And the V region, this V region of the heavy change is encoded by V, D, and J gene segments. And again, in the loci of the immunoglobulins, remember, we have multiple versions of each of these segments, okay? Multiple versions of these, multiple versions of these segments, and multiple versions of J segments. And so these are randomly selected and brought together in each individual developing B cell. They are randomly selected and brought together by DNA gene rearrangement to determine, again, the amino acids that will be forming the variable region of the heavy change and variable region of the light change. And so random recombination of gene segments produce diversity in this antigen binding site, again, that is determined by the variable region of the heavy change and the variable region of the light change. And so this is one of the ways in which this diversity of the amino acids in this antigen binding site will determine diversity in the specificities of the developing B cells that are being generated all the time in our bone marrow, okay? Because in each B cell, these segments, the version of these segments one of these versions of V, one of these versions of V, one of these versions of J, in each B cells will be randomly selected. So will be different than in the neighboring developing B cell. And so that's why these two B cells that are developing in the same bone marrow may select different B, different D, different J's, and ultimately will have different amino acids in the variable region of the heavy change and or the variable region of light change, and so they will have different specificity, okay? And so you can see all the combinations you can have here of different B's with different D's with different J's will give you millions of combinations that will give you millions of possibilities for amino acids in the antigen binding site of the developing B cell BCR, and will give you millions of specificity. So all cells have this conformation in their immunoglobulin locus. So even a hepatocyte has this germline conformation. So a hepatocyte, if you look at the loci of immunoglobulin in a hepatocyte, you will see exactly this. You will have many B's, many A's, many D's, okay? Same as it is depicted here. This is the germline, okay, before it has been recombined. And it's in every cell in our body, we have this conformation in the immunoglobulin locus. However, in the developing B cells, and only in the developing B cells, there will be these rearrangements of the immunoglobulin, again, where particular versions of the B's, of the D's, and their J's are brought together to create functional immunoglobulin light and immunoglobulin heavy change genes. And so this is one way to illustrate that is like, imagine that you have a full course, right? Like that you are in a big room now, and I pick the hair from one student, the eyes from another student, the nose from another student, and the mouth from another student. And this would be the face or BCR of one developing B cells. Then the next developing B cell that will pick the face, the hair from another student, the eyes from a different student, the nose from the student in the back, and the mouth from the student in the front, and will form another face or another BCR, because it combines different pieces of this face, or combine, in the case of BCR, different segments, versions of the segments that together encode the BCR. So is there any question? So by randomly picking different pieces that encode the overall face, at the end, I will have many versions of the faces in the room, right? And the same thing, the developing B cells, at the end, we have many versions of variable regions in the heavy change and the light change that will determine many specificities of the BCR. Is that clear? Any question?
I have a question.
Yes, Madison. Yes, please.
What does the purple box encode for? Because in the previous one, I noticed that the purple box isn't spliced out, but it doesn't show up in the protein.
These are all encoding for the constant region of the immunoglobulin M. And so constant region of the heavy change in this case is the immunoglobulin M, because it's C-mu. But we will get to that a little bit later about how the constant region is brought together and is selected to induce different isotypes.
Okay, thank you.
Sure, thank you. Okay. So how this gene recombination works? So the RAG proteins will bind to sequences that are called RSS that are basically flanking the versions of the segments that need to be recombined. And these RAG proteins are nucleases that basically will break the DNA at the joints. And so RAG stands for recombination activating genes, and are special recombinases that only are expressed in lymphocytes during very specific developmental stages, okay? So only in B cells, and we will see also this the process is very similar for the TCR, which we will learn later. But so these RAG proteins play a similar role in the TCR recombination. And so these RAG proteins are only expressed in developing B cells and T cells, so only lymphocytes at very specific developmental stages. And so that makes sense. And that is because in all the cells, we have the germline loci configuration, but only in lymphos, in B cells, we have rearranged the immunoglobulin. So RSS stands from recombination signal sequencing, and is the unique signal sequence that direct these recombinations by the RAG proteins. And so here, you can see here the RAG protein will bind to the RRS sequences and it will break the DNA, and that will release or excise this intermediate segment of DNA, and will bring a V and a J segment together, okay? And so other enzymes that you don't need to remember the names will help in joining now the V and the J to form this coding joint, okay, of the particularly selected V segment. In this case is V1, and the particular selected J segment, which, you know, could be J3, let's say. And so now this will encode the variable change of the variable region of the light change. And this signal joint will be degraded, and there's no known role for it, so forget about it. But you need to remember that here there is a coding joint that has been formed after the excision of part of the immunoglobulin loci. Okay. So when this occurs, when there is the cut by the RAG proteins, and there are other enzymes that favor these joints, this creates another mechanism for increasing immunoglobulin diversity that is called imprecise recombination junction, because within this joint, there will be addition and excision of nucleotides that have no template, that will be different because they have no template. It will be different in different cells, and it will be necessary for bringing together these two segments. And that adds additional changes in the sequence of the DNA that will be different in different developing B cells, which will increase further the diversity. And so here, this is represented here, the imprecise recombination junction. So the DNA is clipped to create a hairpin structure at the end of the immunoglobulin segment. So this is a B segment and a J segment. And so the RAG proteins will cleave here, And so other proteins, and again, I don't need you to remember the names, will bind to the hairpins and further cleave the RRS. And here I want you to remember this part. So the DNA hairpins are cleaved at random, but then there are additional bases that may be added by this enzyme that I want you to remember the name, that is terminal deoxynucleotidyl transferase, or TDT. So this is the enzyme that will be adding nucleotides at these imprecise junctions. And also there will be some subtractions of nucleotides by exonucleases that will generate at the end these imprecise ends that then are brought together with DNA ligases to form this coding joint, okay? But you can see what I think that what I want you to remember is that these TDT enzymes are important for adding segments here after the RAG proteins have cleaved the DNA. And I want you to remember that this addition of an excision of nucleotides is random. So that means that in one developing B cells, you may add certain nucleotides that will be different than the ones that will be added here in the coding joint in the next developing B cells, even if they happen to have selected the very same B and the very same J. This junction will be different because occurs randomly. And so we learned in the previous lectures what are the CDRs, and that the immunoglobulins in the variable regions, they have 3 CDRs or hypervariable regions for the heavy change variable region or for the light change variable region. And I mentioned to you, and I promise I will explain why, that the CDR3 has the highest variability. And the reason why is that the CDR3, so the amino acids here in this hypervariable region 3 or CDR3, they happen to be encoded by part of the variable region that includes 2 joined coding joints, okay? And so you can see the CDR1 and the CDR2 happen to be encoded by the V segment, but the CDR3 happen to be encoded mostly by the D segment in the heavy change, but also contain the junction between D and V, and also the junction between D and J, right? And so what I want you to remember basically is that they contain 2 junctions. 2 of these imprecise junctions that I told you will add diversity in the amino acids of developing B cells, because the amino acids here are added and excised without a template, and so they are randomly added. And so this makes this the CDR3 having 2 encoded by 2 imprecise junctions more diverse in their amino acids and having higher variability in the pool of immunoglobulins. Okay, is that clear? Okay, thank you. Okay, so I see some fingers up. Okay, so let's see, because this can be a little bit abstract. So I decided to actually have this movie during the lecture. It's a 2-minute movie, okay?
Immunoglobulin genes are composed of...
Let me know if you cannot hear well, okay? I think that it will be fine, but let me know if otherwise.
Immunoglobulin genes are composed of separated segments of DNA that become joined together by a process called somatic recombination to make a functional gene. In heavy chain genes, there are 3 gene segments: the variable or V segment, a diversity or D gene segment, and the joining or J segment. Light chain genes, such as those shown here, have only two gene segments, the V and the J segments. Gene segments that can be recombined have specific sequence motifs adjacent to them, called recombination signal sequence, or RSS motifs. A protein complex containing the products of the recombination activator genes, RAG1 and RAG2, binds specifically to the RSS motifs. In this example, flanking a V gene segment and a J gene segment. The individual gene segments, to whose flanking RSS motifs the RAG protein complexes bind, are selected at random from a number of copies present at each gene locus. The RAG protein complexes bring together the gene segments to be recombined and cleave the DNA exactly at the junction of the gene segment and its adjoining RSS motif. The cleavage creates a hairpin of DNA at the end of the gene segments and double-stranded breaks at the ends of the RSS motifs. Additional proteins, DNA-dependent protein kinase, COO, Artemis, and a dimer of DNA ligase and XRCC4 are incorporated into a large complex with the RAG proteins. These RSS ends are joined, forming what is called the signal joint, to create a closed circle of DNA that plays no further role in the recombination process. The DNA hairpins at the ends of the gene segment are cleaved. An additional enzyme, terminal deoxynucleotidal transferase, or DT, is recruited and adds additional nucleotides to the ends of the DNA strands. The other enzymes in the complex ligate together the two ends of the gene segments, completing the recombination process.
Okay, so again, I don't need you to remember all the names of the enzymes, so just remember the RAG proteins are important for the cleavage and together they act together with other proteins, and the TDT is important to add randomly amino acids in the joint, increasing the diversity by the second mechanism, that is the imprecise junction junctions. And so the third mechanism is the combinatorial pairing that will further increase a diversity of the immunoglobulin variable region. So because each antigen as binding site, as we learned before, is constituted by the variable region of the light chain and the variable region of the heavy chain. Again, each of them is constituted by the variable region of the light chain and the variable region of the heavy chain. Pairing of different immunoglobulin heavy chains with a different immunoglobulin light change adds diversity during the immunoglobulin generation. So let's say that two developing lymphocytes, they generate exactly the same light change. But now this light change will combine with different heavy chains in these developing lymphocytes. And so ultimately the two developing lymphocytes, despite having the same light change, they will have different antigen specificity, because this light change that is the same in this case will combine with a different heavy chains that have been randomly rearranged. So just the combinatorial pairing and rearrangement together, if you do the math of how many variable region versions occurs, so there are, for the light change, there are 70 versions for the V region of the light change, and 9 versions for the J region of the light chain. And there are 65 versions of the V region of the heavy chains. There are 27 versions of the D region of the heavy chains, and there are 6 versions of the J region of the heavy chain. So when you start putting these three together, right, it gives you about 10,530 different versions of the variable region for the heavy chains. And this is the amount of versions for the variable regions of the light chains. and then you can when you start combining these together, the variable region of the light chain with the variable region of the heavy chain, and you see all the possibilities, it gives you about 5 million different antibodies specificities. And that's without the imprecise junction, which again is the second mechanism we mentioned that further increase the diversity because of TDT adding nucleotides randomly in the coding joint. Okay, is there any question about that?
I had a question, Professor.
Yes, Megan.
In somatic recombination, are the gene segments, are they like just kind of ligase together as opposed to imprecise recombination where they have like the RAG enzyme and then other enzymes that eventually form the coding joint?
Okay, that's a good question. I'm glad you add that because this is the same process. It's in the same cell occurring at the same time. So the somatic recombination is picking this different version of B with a particular version of a J. That's the somatic recombination, and bringing them together. And in doing so, in the coding joint that brings the two segments together, there will be imprecise junctions, okay, because there are nucleotides that will be added. So it's only one process, but there are two mechanisms that result in increased diversity. The first mechanism is the somatic recombination because you will be picking randomly different, you will be picking randomly different segments, okay, different versions of the segments. And that's somatic recombination, that will add diversity. But to bring these segments together, you will have enzymes like TDT that will add nucleotides randomly into the coding junction. When you bring these segments that you randomly pick together, this TDT will add nucleotides randomly, and that is the second mechanism that further increase diversity, these amino acids, okay? So the first one, the somatic recombination, is the diversity added when you pick different versions of one of the 65 versions of the V segment that encodes, that are in the loci for the heavy chain, one version of the D, and one versions of the J, okay? So depending on which one the developing B cell would pick to bring together, that will determine diversity of the specificity. And on top of that, when that is brought together, now the TDT will add nucleotides randomly into the joints of these segments. And that's the second mechanism, bringing a specificity, increasing diversity in specificity.
Okay, that makes sense. So just to summarize, both somatic recombination and imprecise recombination are like one process, but then they both contribute to increasing the diversity.
Absolutely, yes. And the combinatorial pairing is part of the same process. Now, once you generated the immunoglobulin light chain and the immunoglobulin heavy chain in the same developing lymphocytes, you need to bring them together to be able to express as BCR in the surface of this developing lymphocyte.
Okay, that makes sense. Thank you. And I had one more question, if you don't mind.
Yes, please.
This is more for curiosity's sake. So when you have the insertion of nucleotides by the TDT, could that cause something similar to like an insertion mutation, or like where it like makes the coding joint non-functional?
Yes, that could happen. That could happen. And basically that will result in expression of a non-functional polypeptide and so no functional BCR. And we will learn in that lecture how the B cells that fail to express a functional BCR, they will die.
Okay, thank you.
Cool. Great questions, Megan. Okay, so all this occurs before the antigen encounter and generates a diversity in the variable region of the heavy change and light change, okay? After antigen stimulation or antigen encounter, there is this process that occurs that we will briefly mention now and we'll explain more later, that is called somatic hypermutation. So this somatic hypermutation only occurs after antigen stimulation and is dependent on this enzyme that is called AID, adenosine deaminase, which will introduce random mutations that are inserted in the variable regions of the immunoglobulin light change and immunoglobulin heavy change. And so, again, this enzyme is the one that will introduce these random mutations in the variable region of the heavy change and light change, and this only occurs after antigen stimulation and does not affect the constant region. So AID is activation-induced cysteine deaminase and converts cysteine residues in the DNA to uracil DNA, and then these uracil residues are excised by another enzyme. You don't need to remember the name of this one, but it's uracil DNA glycosylase, so only remember the name of AID, please. And as a consequence, non-templated nucleotides are introduced at this site. And so that increases further the diversity after antigen stimulation. So the mutation rate of immunoglobulin genes during this process that is called hypermutation is about 1 million times higher than the normal genomic mutation rate. And this process called somatic hypermutation, again mediated by this key enzyme AID, can lead to antibodies with because the introduction of the mutations are random, can lead to antibodies that now have higher affinity for the antigen that they encountered, lower affinity for the antigen that they encounter, or they may change their affinity and start generating affinity to even self antigens or other antigens. And so I'll leave you now with the intrigue, and later we will learn that there is a selection process that is driven by the antigen that basically eliminates those B cells in which the affinity of the BCR has been reduced or have changed to generate affinity to self-antigens, for example. And so I'll leave you with that, and we will learn more about that later in future lectures. Okay, any question about that? Okay, so let's learn now how does isotype switching work. So this is again the loci of the immunoglobulins, but in this case is after the variable region has been rearranged, right? So this is the heavy change loci, that is where the relevant one for isotype switching. And so I mentioned to you before that we have all the segments in the germline. We have all the segments, remember, that all the V's, all the D's, all the J's that encode for the variable region of the heavy change. And then we have the downstream, the constant regions. And so in this case, it's not germline anymore. This is a developing lymphocyte that have already rearranged the VDJ segments. So now once that's happening, now this can be transcribed, this DNA can be transcribed. And these transcriptions initially will always extend the C mu and C delta. And these are, again, encode for the constant region of the immunoglobulin M and the immunoglobulin D. And so you can see here that are other constant regions. The C gamma 3 encodes for the constant regions for the immunoglobulin G3. We have C gamma 1, C alpha 1, C gamma 2, C gamma 4, C epsilon encode for the constant region of the immunoglobulin E, C alpha 2, constant region of immunoglobulin A2. Okay, and so in between these constant regions, not in between C and delta, C mu and C delta, but so these are not separated here by switch regions, but all the rest are separated by what are called switch regions that are key for determining the isotype switching that we'll learn in the next few slides occurs also by DNA recombination. And so in a developing lymphocyte, once the BDJ are rearranged, now the DNA can be transcribed, and is always being transcribed first by to extend to the C mu and C delta. So here is the DNA form with C mu and C delta. These are the different exons and introns in here. And this will be transcribed to generate a messenger RNA that encodes for the variable region with the selected BDJs, right? And the C mu and the C delta, okay? This is the messenger RNA now. And so the naive B cells will express both IgM and IgE only in the membrane-bound form, and we'll learn more about that. But now focus on this. Once the messenger RNA that encodes mu and delta is generated, now alternative messenger RNA splicing will determine if C mu is the one that will be kept in a mature messenger RNA that then will be translated into protein and to generate surface immunoglobulin M in these developing lymphocytes. Or in some cases, even in the same developing lymphocyte or in the same cell, the alternative messenger RNA may splice to generate a messenger RNA, mature messenger RNA that now encodes for the C delta, and it will generate a polypeptide, a BCR, that has the very same variable region here, right? Because it was the same messenger RNA that we were talking about in the previous slide. Here you can see is the same variable region that is kept by alternative splicing brought together with the C mu, or the same variable region that by alternative splicing is brought together with C delta. Okay, when it's brought together with the C delta, it will generate a BCR of the immunoglobulin D isotype in the surface of these B cells. Okay, and this can occur in the same B cell. So the same B cell can express both IgM and IgD. But in both cases, this BCR that this B cell is expressing, the IgM and the IgD that is expressed of the very same B cell, will have the same variable region here, right? And so will the specificity be the same? Maybe we can do, if you think the specificity will be the same, then press yes. If you think the specificity will be different, then press no. Let's see. If again, this can occur in the same B cell, but that some messenger RNA will mature to generate IgM, and some messenger RNA will mature to generate IgD. And so now this B cell would express both IgM and IgD, but the specificity will be the same of this in this very same B cell. The specificity of the IgM BCR and the IgD BCR in the very same B cells will be the same. Press yes, or will be different. Press no. Okay, so whoever voted said yes, and that's the correct answer. So, and the reason why the specificity will be the same is because during this splicing, the variable region is not touched. It's the same variable region, the same BDJ that have been rearranged before at the DNA level are now brought together with the messenger RNA that encodes for C mu or messenger RNA that encodes for C delta. So the naive immature B cell make only C mu. So at the B cell that is maturing in the bone marrow will only to generate IgM. And IgM is the only immunoglobulin that will be expressed in the immature B cell that is developing in the bone marrow. And then when this immature B cell will exit the bone marrow and will mature, and when matures, it can start splicing a part of this messenger RNA into IgD, and another part will continue splicing IgM, and will then express both IgM and IgD in their surface with the same antigen specificity, because there is the same variable region in both cases. Okay, so, well, okay, maybe this is kind of easy, but let's do this poll question. Okay, so I will end the poll, and I'm glad that I asked this question, because it seems that, you know, needs further clarification. So the isotope switching I see. I think let me explain the next few slides, and then we'll do the poll question again. Okay. So what I was mentioning here at the messenger RNA splicing is how it's not isotype switching. It's what occurs before antigen encounter, and is to determine whether IgM or IgD is expressed before our antigen encounter. The isotype switching is occurring after antigen encounter, only if T cell help is received by the B cells. And so remember, these lectures will learn some CD4 T cells become effectors. They may acquire the capacity to help B cells, and these are the subsets of CD4 T cells that are called T follicular helper cells. And so they will help the B cells that are presenting the antigens for which the T cells are specific in MHC class 2, right? And so these B cells has received the BCR stimulation, but also is receiving T cell help. And what this T cell help does is increase the expression of, again, this AID enzyme that is also essential for DNA recombination, that now at the DNA level will bring together different switch regions to determine which is the constant region that will be transcribed, okay? So this is here, before on the top, what we were talking about before is before antigen encounter, IgM and IgD can be produced, okay? And so the transcription expands always till IgM and IgD, and then RNA splicing determines if it is only IgM or if it is both IgM and IgD in a mature naive B cells. So now this naive B cell that has this configuration on the top, if it receives T cell help, it will upregulate the expression of the enzyme AID. And that will promote DNA recombination, again, affecting the DNA configuration to bring these switch regions together in a way that will now bring this very same variable region. You can see here the variable region is untouched, but now we'll bring it closer to a particular constant region. In this case, is the constant region encoding for the alpha-1. So this now B cell, after this isotype switching rearrangement at the DNA, will produce the very variable region, so the same specificity with the constant region alpha-1, so immunoglobulin A1 with the same specificity as it has before seeing this antigen, right? Because again, you can see here the variable region didn't change. So the IgA1 will be produced in this case after the switch. So the antigen stimulation that occurs without B cell help, so in this case, the B cell may not have received, might have seen the antigens, may not have received T cell help for whatever reason, and we will learn more about that. And now these B cells will continue with this configuration, but now will be activated, and in some cases can differentiate into plasma cells that produce IgM without any isotype switching, because there was no T cell help, there was no upregulation of AID, which is essential for this switch region recombination that is the isotype switching. Okay, so let's do now again this poll question. Okay, I will end this. So most of you now understood the correct answer is A. So how does isotype switching affect the affinity of the BCR? The affinity of the BCR for the BCR antigen binding site for the antigen. So the affinity and the specificity of the antigen are determined by the variable region of the immunoglobulin. And we just said that the isotype switching affect the variable region, will recombine these switch sequences here that are the ones that will bring different, will excise the C mu, the C delta, and maybe other constant regions to bring together particular, particular constant regions in close proximity to the very same variable region encoding genes segments, okay? I also want to clarify that what I mentioned in the previous slide about this how the expression of IgM and IgD are determined by RNA splicing, that's not isotype switching. They can be both produced together. That's how, just so you learn how IgM or IgD expression is determined, but it's not isotype switching because it doesn't involve the switch regions in the DNA, which occur after antigen encounter, okay? Okay, any question?
Professor?
Yes, Brian.
On that slide that that you just showed with the T cell help, can you go back to that one?
Yep.
Oh, wait, no, before.
This one?
No, I think it was one of the last slides. It's the one where you talk about like antigen encounter. That one, yeah.
Yep.
For that last bullet point, where you say antigen stimulation without B cell help, why is it B cell help or T cell help?
It should say T cell help. Thank you for
okay.
I will correct, or you guys, is a small correction, so please make the correction. Thank you, Brian.
Of course, thank you.
Yes. Okay, so let's go through the last topic. It's very brief. How do B cells secrete their immunoglobulin? So we learned that B cells secrete their antigen receptor without the transmembrane to produce these antibodies, okay? And that this occurs after the naive B cells become activated and differentiate into effector B cells, that is these plasma cells that now will produce the antibody. And I just want to give you some details at the molecular level on how this occurs. Again, the surface and secreted forms of an immunoglobulin are derived from the same heavy chain gene by alternative RNA processing. So this is the transmembrane immunoglobulin M, and so it has this variable region, the constant region for IgM that is transcribed. It's brought together in the mature messenger RNA to the variable region, and also will be transcribed the transmembrane region. When the B cells switch to become a plasma cells, now there is further RNA processes that is a different one that now brings together leaves out the transmembrane regions, and so this immunoglobulin can be secreted. But you can see that's the only part that changes when the effector B cells in this process become a plasma cell. The variable region that determines antigen affinity and specificity is the same. The constant region of the BCR is the same as the constant region of the immunoglobulin that now is secreted by the plasma cell. And so, and this switch, or it's not switch, but this process in which the transmembrane part is left out for the immunoglobulin to be secreted occurs also at the RNA level. Okay, so let's go through a brief summary here, and then we'll continue next lectures with the developmental stages of the B cell and how what we learned today fit into that. So we learned that there are 4 mechanisms to generate antibody diversity, particularly at the variable region of the immunoglobulins that determine the antigen specificity and affinity. We learned these are the somatic recombination that is given by the existence of different versions of B, D, and J segments that are brought together to generate a combination of segments that encode for the variable region of the immunoglobulin. And in bringing those together, there is the generation of imprecise junctions. That is the second mechanism that increase the diversity, and is the reason why the CDR3 have a higher hypervariability in the amino acids. And the third one is the combinatorial pairing, that is the pairing a particular light change with a particular heavy change that will further increase diversity. And this 1, 2, 3 occurs before antigen encounter. And we learn that after antigen encounter, there is further hypermutation again within the variable region of the immunoglobulins that is mediated by the same enzyme that participates in the switch, that is this AID enzyme. Okay, and we will learn how this occurs, but these are random mutations that occur after antigen encounter, and we just mentioned that it's a selection process to make sure only the higher affinity resulting BCR after the somatic hypermutation are selected. So we also learn how does isotype switching work. And so we first learn that before antigen encounter, a naive B cell only makes C mu or C delta, okay? And basically, depending on whether the B cell is immature or is mature, there will be a particular RNA splicing that will allow production of C mu only in immature naive B cells, or both C mu and C delta in the mature B cells. But this is all before antigen encounter, and this is not what we call isotype switching. What we call isotype switching occurs after antigen encounter, and this isotype switching occurs via DNA recombination of switch sequences that are flanking the constant regions of the immunoglobulin heavy change, and occurs when AID is induced via T cell help. And last, we learn how do B cells secrete their immunoglobulin, and we learn that when a B cell becomes an effector cell or plasma cell, there will be further RNA processing that will occur, that the only thing we'll do in this eliminate the transmembrane region, but will keep the same constant region and the same variable region of the BCR that this B cell was expressing. Okay, with that, I think that we can end this lecture.